welcome. So, today we will be going ahead with Biotechnology Principles and Processes Part 4, which is transformation of rDNA into host. We have talked about the introduction, we have talked about the tools for rDNA technology, where we have talked about restriction enzyme, cloning vectors, and multiple cloning sites. Now, the last but the most important part is I need to have a host that I have already told you in my previous video. So now, once I have a multiple cloning site, these cells needs to be transformed into a host. So when I talk about hosts, they are the vectors for cloning genes in plants and animals. So, in plants, if I talk about, there is Agrobacterium tumefaciens, which is a pathogen of dicot plants which deliver tDNA, which transform normal cells into a tumor. So, this tumor inducing plasmid, which we call it as Ti plasmid of Agrobacterium, is now modified into a cloning vector. So now this particular TI plasmid is going to induce tumor in Agrobacterium tumefaciens in the plants. Other vectors we have retroviruses, which is used in animals that transform the cells into cancerous cells. And now they are disarmed and used as cloning vectors. If I talk about the competent host, for transformation formation with recombinant DNA, the first option that we have, simple chemical treatment with the divalent calcium increases the efficiency of host cell to take up the rDNA plasmid, recombinant DNA plasmid. So this recombinant DNA that we have, they are just resuspended in calcium chloride and incubated on ice. So once they are incubated on ice and they are swirled or they're centrifuged, then you can wash the cells and resuspend in transformation buffers. I have the transformed cells there. The other method is what? Other method where the recombinant DNA can also be transformed into a host cell by incubating both on ice followed by placing them briefly at 42 degrees Celsius. This is known as heat shock, and then putting them back on ice. So you put them on ice, then heat it again, and then put them in ice immediately. This enables the bacteria to take up the recombinant DNA. There is another method which we call as microinjection. In microinjection, the recombinant DNA is directly injected into the nucleus of animal cells. So there is an injection wherein I have the recombinant DNA. I will push the recombinant DNA into the nucleus of the host cell. Another method we have is a particle gun method. So there is a gene gun, or I tell it is a ballistic particle delivery system designed for plant transformation. It's a device for injecting cells with genetic transformation. The payload is an elemental particle of heavy metal, tungsten or gold, called microprojectile coated with plasmid DNA. So I have a gene gun in a plastic disc with DNA coated gold particles are there. So the disc is topped by a screen, and when it is being pushed in, this DNA coated particles will spread to the target plant cells. So I can go ahead, and this is a particle gun method. Then I can also have a disarmed pathogen. So first I told there is a physical way of, a chemical way of inducing the transformed DNA, wherein there is a chemical treatment with calcium chloride. Then I have a heat shock manner where I have put it in ice, then give a heat shock and then put it in ice. Then there are physical methods like microinjection, particle gun method, and there's a biological way where I am using a disarmed pathogen that I was talking to you earlier about. Pathogenicity, when I say pathogen means it will create, it is foreign and it is infectious microorganisms that can cause disease or sickness. When I'm disarming the pathogen means I am removing the pathogenicity of the vector. Pathogenicity is the reason, it is the reason why this particular pathogen is causing a disease. So disease causing factor is being removed, so that is why the arms it is going to carry, the guns, or whatever the harmfulness it is going to carry is being removed. They are being disarmed. So this disarmed pathogen, it will allow us to infect host and transform the rDNA. Can be Ti plasmids in case of plants, can be retrovirus in case of animals. So here we have talked about all the tools which is necessary for the transformation, okay, for producing rDNA. I have already talked about restriction enzyme. With the restriction enzyme you cut the cell, then after that you are ligating it. Once you are ligating, so restriction site, we have selective markers for it to replicate. I have ORI. So for replication there is an ORI which maintains the copy number of the cells. I have selective markers that there are two kinds of selective markers. There's antibiotic resistance as well as laxi. So there I can screen out the transformed cell and then I can transform it into the host. So that's it. Thank you.